Hello all! This week we will go back to Chapter 2 and talk about the brain and behavior. This chapter may be a review, especially for those who have taken anatomy and physiology courses. In Part 1, we will discuss about nervous system, structure of a neuron, the function of a neuron, how do neurons communicate with each other using neurotransmitters, and the different types of neurotransmitters. So, a nervous system is like an extensive network of cells that carry information to and from all parts of the body. It's like if you're using Facebook, Instagram, or any other social media, all the users out there are your friends and you're constantly communicating with each other. And that's pretty amazing, but probably annoying at the same time. But that is a very important thing to our nervous system. And neuroscience is a field that studies about nervous system, and with the advancement of technology, these fields are becoming more and more popular. So there are two types of cells in a nervous system. A neuron is one of them, and the other cell is called glial cell, which we'll talk about it later. Neurons look like this, and it is made of pretty simple structure. Neuron handles the information processing function, and so neurons are again constantly communicating with each other. So neurons can be broken down into these basic parts. Dendrites are tree-like structures that receive messages from other neurons. Cell body is where it contains nucleus, which is responsible for maintaining the life of the cell. It's a pretty important part of the neuron. An axon carries information to other cells or other neurons. So again, neuron is a very basic, simple structure. And you might be wondering, what are those stuff around the axon that looks like multiple sausages? And that is the myelin sheath, which contains glial cell. Do you remember what we briefly discussed about glial cell in the beginning? So we talked about there are two types of cell in the nervous system, and one was the neuron, and the other was this glial cell. So what are the purpose of the glial cell? So it has several functions. And one includes to protect the neurons, and the other is to speed up the neural impulse. So, what is a neural impulse? This is how neurons communicate with each other, basically sending messages to one another. And it uses an electrical impulse. So in a way, we are like batteries and have positive and negative. And some parts of the neurons are more positively charged and others negatively charged. And there are specific terminologies to represent these states. So again, one of the primary purposes of the neural impulse is to send messages through the axon. And when the neurons are negatively charged and inactive, we call that state as resting potential. However, when the neurons are going to send a message, then actual potential occurs, which is a brief wave of positive electrical charge that sweeps down the axon, and this is the driving force that pushes the momentum forward. So let's take another look of a neuron, like in this figure. So as you can see, our neurons are charged with either positive or negative, and during the resting potential, when there are no action, inside of our neurons are negatively charged, approximately around negative 70 millivolts, whereas the outside of the neurons are positively charged. So, to explain this phenomena in detail, there is first the cell membrane, there it is, which separates the outside and inside of the neuron. And then there are several ions and molecules that comes into play. So first, there's a potassium ion, which is positively charged, and then there is a sodium ion, which are also positive. And then there are chloride ions, which are negatively charged. And last, there are protein molecules, which are negatively charged. And negatively charged protein molecules cannot pass the membrane. But potassium ions are able to go back and forth between inside and outside of the neuron. just like this. And this represents the resting potential. So you kind of see that inside the cell there are more negative ions molecules. And we will use this figure to keep track of whether inside the neurons are negative or positively charged. So again, during the resting potential, the inside of the neurons are negatively charged at about negative 70 millivolts. But when the neurons are ready to pass on the message, then action potential happens, creating the wave of positive electrical charge that sweeps down the axon. But notice that as soon as this wave passes by, the inside of the neurons would immediately go back to a negative state again. So using this figure again, what happens during action potential is that the sodium channel opens, which allows the sodium ion to rush inside the neurons. And again, making the inside of the neurons to be positive. So the sodium ion enters inside the neurons, making it positive. And we call this depolarization, as the inside of the neuron is no longer polarized as negative. And now the inside of the neuron is about positive 40 millivolts. But as we discussed, right after the action potential, the neuron goes back to negative. And this is done as the potassium pump opens, allowing the potassium ions to go out of the neuron. Which makes the inside of the neuron negative again. And now we're back to where we began. And this cycle just repeats as the neuron attempts to communicate with each other. So as the inside of the neuron goes back to negative, we call that as repolarization, as inside of the neuron is once again polarized to negative. And notice that there is a dip and that goes beyond negative 70 millivolts. And this is called hyperpolarization, which is caused by an overshoot in the movement of potassium ions. And so now we've covered the cycle of neural impulse. So with this neural impulse, there is one important principle, which is called the all or nothing principle. This basically means that a neuron either fires completely once it hits the threshold of around negative 55 millivolts, or does not fire at all. So in other words, once the inside of a neuron becomes negative 55 millivolts and action potential happens, then there is no going back. And as we discussed, after the action potential, it will go back to the resting potential. And one interesting thing about the action potential is that the intensity is the same, even when someone taps on a shoulder, for example, or even when elephant steps on your foot. But what's different is that the frequency of action potential, where if an elephant steps on your foot, the number of times the action potential happen is greater than as opposed to someone just tapping on a shoulder. So as the action potential carries the electrical impulse down the axon and to relay the message to other neurons, notice that there are tiny gaps between the neurons. And this gap is called synapse or synaptic gap. So how do neurons communicate if there is a gap in between? So this is what happens. As we discussed, the action potential sends message to the tip of the axon and at the terminal button, which is, again, at the tip of the axon. And there are what's called a synaptic vesicle, which is like a sac, and inside there are neurotransmitters, which are like chemical messengers. So with the force of the action potential, it pushes the neurotransmitters to the synapse or the gap. And the dendrites, or the receptor site of the receiving end of the neuron, will capture these neurotransmitters, triggering the next action potential. But note that there are what is called excitatory and inhibitory neurotransmitters. Excitatory neurotransmitters make an action potential more likely to happen after the dendrites of the receiving end of the neuron captures these neurotransmitters. But the inhibitory neurotransmitters make an action potential harder to achieve. And there are literally at least 100 different neurotransmitters. And I'm pretty sure you want to know all of them, but in this video clip, I will talk three of them. But before we dive in discussing the specific neurotransmitters, I want to discuss what happens to the neurotransmitters once they are released in the synaptic gap, because they are not always received by the receptor sites of the receiving end of the neuron. They can be reabsorbed by the sending end of the neuron, which is called a reuptake, or it could be deactivated by an enzyme. And in this video clip, we will focus on number 2, where the amount of neurotransmitters to be released may be inhibited or activated, and number 4, reuptake, where the neurotransmitters are taken back. So the first neurotransmitter we will talk about is dopamine, which controls the voluntary movement and influences attention, mood, and sleep. As a result, a lack of dopamine is known to be associated with Parkinson's disease, where the physical movement deteriorates, but too much dopamine can also cause symptoms of schizophrenia, like symptoms of hallucination. So basically, drugs, both legal and illegal, attempts to target specific neurotransmitter and enhance or reduce the effect of that neurotransmitter. For example, cocaine acts by blocking the dopamine reuptake sites, causing the dopamine to float along the synaptic gap longer and accumulate, which would increase the effect of dopamine. So do you remember what happens if there are too much dopamine? It resembles the symptoms of schizophrenia, such as hallucination, which is why cocaine causes individuals to see things that others cannot see or hear things that others cannot hear. Another neurotransmitter is called acetylcholine, which is an excitatory neurotransmitter, and it is involved in actions of muscles, learning, and memory. As a result, a lack of acetylcholine is associated with Alzheimer's disease, which is a brain disorder that involves decline in memory. And one substance that targets acetylcholine is the black widow spider venom, which stimulates release of acetylcholine. Right here. And because the wastage of acetylcholine is known to cause, for example, muscle pain, difficulty breathing, and muscle cramps, we can think of too much of acetylcholine as related to an excessive movement of our muscle. Another neurotransmitter we will discuss is serotonin, which is mainly inhibitory and regulate sleep, mood, and attention. And lack of serotonin is known to be associated with depression. So drugs like Prozac, Zoloft, Lexapro are known to be selective serotonin reuptake inhibitors, or SSRIs, which basically blocks the reuptake of serotonin, making serotonin last longer in the synaptic gap and increasing the influence of serotonin. So, as we just covered, a drug could either enhance or reduce the effect of neurotransmitters. Everything we talked so far, like cocaine, black widow spider venom, and SSRIs, can be thought of an agonist because they all enhance the effect of specific neurotransmitters. For example, black widow spider venom was enhancing the effect of acetylcholine, and SSRIs were enhancing the effect of serotonin. So, an example of antagonist is Botox injection, which are used for cosmetic to stop muscles from moving or dropping in order to prevent wrinkles. And we briefly talked that lack of acetylcholine causes paralysis. So Botox injection reduced the effective acetylcholine and therefore an example of antagonist. So hopefully you understood the nervous system, the structure of a neuron, and how they communicate, and different types of neurotransmitters. And if you have any questions, let me know. And otherwise, I will see you in part 2 of the video. Thanks and take care.